Hi, I'm Nurry Hong, and welcome to the Innovation Series. This is a series where we're going to talk about everything related to the science of food allergy and everything new and up and coming. Last time we had a conversation with Cynthia Rice from JDRF, and we talked about the state of prevention in type 1 diabetes and the role that nonprofit institutions can play in helping to drive innovation. Today, we're going to return back to a topic that's central to food allergy. We're going to talk about food allergy science and the causes of food allergy, loss of tolerance, or the gain of tolerance. Today, we have the privilege of welcoming Dr. Cecilia Berin. Hi, Cecilia.
Hi, Nurry. Thanks very much for inviting me on.
So before we begin, I'm going to give a little bit of a proper background of yourself, and then we're going to get into all things food allergy science. But I wanted to also start with just a little bit about what you're doing and how your research is. Cecilia is the Endowed Chair, the Hugh A. Sampson Professor of Food Allergy Research. She's Professor of Pediatrics and Deputy Director of the Jaffe Food Allergy Institute at Mount Sinai. Her laboratory studies immune mechanisms of allergy tolerances to food and early life susceptibility to allergic disease. Her research has used models of food allergy to identify mechanisms of sensitization to foods and to study the communication between the skin and the gastrointestinal tract in food allergy. She has also designed mechanistic studies for multi-center clinical trials for the NIAID-funded Consortium for Food Allergy Research, or COFAR, and the Immune Tolerance Network, ITN, and also going forward for FAIR and the FAIR Clinical Network as well. So did I miss anything, or is that a proper background?
I think that covers it.
So that's a great, introduction, I think, to what we want to talk about today, which is really the core science of what do we know about food allergy, and why does food allergy even happen, and how do we potentially regain tolerance or mechanisms of tolerance? And we'll talk about what that even means in terms of this, magical world of tolerance relative to cure and some of these other words that are thrown around in food allergy or allergic disease.
Sure.
So that's all, I think what we'll cover today. But before we do that, I want to talk a little bit about you. So how are you? How's everything going?
Great. Things are busy again in the lab, and we diverted a little bit for COVID research, but now we're 100% back to food allergy.
That's great. And I assume, given your role, you have been vaccinated?
I have, which is a huge relief. Very grateful for that.
Fantastic. Before we dive into some of the science, because obviously you are at I think really the core of what's most interesting within the world of food allergy, thinking about early life development and fundamental mechanisms of what causes food allergy, what happens when we are treating food allergy, and what does that mean potentially for the future as we think about different interventions and cures. And before we dive into that, though, how did you even get into this area of research? What drew you to this field of immunology?
Well, I started out studying gastrointestinal physiology, and I was very interested in understanding when things go wrong in the gut. And there are a couple of diseases where that is really at the forefront. One of those is food allergy. The other is inflammatory bowel disease. And I started out working on inflammatory bowel disease and started to transition to food allergy. And I just found it so fascinating that there are so many very intricate immune mechanisms that are trying to balance keeping us tolerating all of the foods that we eat, and still the immune system has to be there to protect us from the pathogens that we accidentally consume.
That's okay. So I'm glad that you mentioned inflammatory bowel disease, or these disease conditions often also known as ulcerative colitis and Crohn's disease, which people may have heard of. And those are obviously related to the GI system and inflammation in the GI tract. And I'd love to maybe come back to some learnings from those fields of research and what are the overlaps, and not just in those disease areas, but in some other diseases that involve the GI and these mechanisms that you alluded to.
Sure.
Okay, and before we do that, I wanted to dive into, so based on what you just said, what do we know about early life development? But I have to say, I don't know if you remember, but one of the last times I think you and I actually met in person, maybe it was the Gordon conference in Ventura. So before that, I think was in Florence at a PAM meeting, perhaps.
Mm-hmm.
And we sat at a table at a dinner, I forget for what, in one of these big castle, dinner ballroom areas. And we ended up talking about the microbiome and the role in early life development, this topic that we're about to dive into. And I said, one of the thoughts that I had with Wayne Schreffler was that we should start a farm service for early expecting mothers and their newborns. So they should all come
Yes,
onto the
I
farm
do remember
day one
this.
and live on the farm while they're, in early stages or in pregnancy and through early life of, for childhood. Just again, in the nature of the microbiome and what's going on with our gut. So with that
I think it's a
segue,
fantastic idea.
by the way, I didn't do the farm idea, but I am now pursuing opportunities in the microbiome. So this is something near and dear to me. What is going on with food allergies? We all know that there's this tremendous rise in the incidence of food allergy. We've all heard of the role of the microbiome and what's going on in the gut. And we all understand at a high level, there's an association there that is just observed here, but more broadly.
Yeah.
But beyond just that, what is going on and what role does that specifically play, or what do we know so far?
Yeah. So, I think that this started out with this observation that there were certain lifestyle factors that were associated with either protection from food allergy or elevated risk for food allergy. Some of those were things like having a dog in the home in the first year of life was protective for food allergy. Growing up on a farm, having older siblings, going to daycare, whereas things like C-section birth were slightly more likely to lead to food allergy. And what all of those things have in common are that they affect the composition of the bacteria that live in our gastrointestinal tract, so our microbiome. Those changes in the composition of the microbiome really profoundly affect the immune system in our gut. So you had, mentioned inflammatory bowel disease. This was what I got my start studying in. And that was one of the first areas where we really started to understand that the microbiota was incredibly important for driving the immune tone of the gut. We've known now for many years that models of inflammatory bowel disease are completely dependent on the microbiota being present. Now we know this from mouse models of food allergy, that the types, not just the presence of bacteria in the gut, but the types of bacteria that are present in the gut are really crucial. And so studying different populations, for example, farm cohorts where there's a very low prevalence of food allergy can give us some ideas. So studying the microbiome of those can give us some ideas about which bacteria may be protective, and importantly, why are they protective? So that we could maybe give back not just those bacteria, but maybe the metabolites that are produced from those bacteria, something simple like a small molecule metabolite that could make our gut immune system more regulatory, more healthy.
I see. And in that case, where a metabolite, for to contextualize, people may have heard of butyrate. Butyrate is a metabolite made by the microbiome and often found in pro or anti-inflammatory responses in many disease areas, including food allergy. And it's something that we've talked about quite a bit in the area of some of these, what they call short chain fatty acids that may be involved. Let's come back to that in a second. I realized as we get into this, maybe it's helpful to step back and to talk about when in early life, what actually happens with normal development and acceptance of food versus what happens in food allergy, which is what we call a loss of tolerance. Could you explain what that means and what's happening there?
Sure. So oral tolerance is a process where when we are exposed to proteins through our gastrointestinal tract, the immune system becomes trained and it becomes non-responsive to that protein that's in those foods. That does not happen in food allergy. And in contrast, there's this process of sensitization that happens. So there are different types of antibodies. So probably we're all familiar with antibodies now from hearing all about it in COVID. But of course, there are these IgE antibodies that mediate allergic reactions. There are different types of antibodies called IgA antibodies and IgG antibodies that are involved in oral tolerance. And so normally on exposure, there is this generation of this healthy antibody response. What's really, I think, interesting is that most children who, have peanut allergy, they will have reactions on the very first exposure to peanut. So there is a process that is happening prior to them reacting to the food where they start to generate those IgE antibodies. And that happens very early in life. And it's a little different for different foods. We know that the first 5 months of age are really crucial for peanut allergy. Allergy to egg and milk may happen even earlier than that. So there's this very early window in life where the immune system starts to generate IgE antibodies to those foods. And there isn't the introduction into the diet to generate oral tolerance. So there's both an active process of generating allergy, making IgE antibodies, and the absence of that regulatory exposure that's going to help suppress those inappropriate IgE antibodies.
Okay, so a lot to unpack there. I just want to pause on a couple of and point on a couple of things because I think you use some terms that people are very familiar with, but to contextualize this, starting with those antibodies. And by the way, isn't it nice that people maybe know more about immunology now? Not to say that COVID was nice, but understanding what a T cell is. And an epitope in the case of antigen presentation. So in the case of our epitopes for, let's say, peanut antigen, what is happening is at some point you lose this ability to use a word you just used, regulate the exposure to that food product, in this case, peanut antigen. And that loss of that regulatory process leads to this antibody development. And we all know what those antibodies are. IgE is the antibody we don't want in food allergy. And for a long time, we've been looking at how do you stop IgE as a method of intervention. And there's obviously a significant value in that. But upstream of that, the research that we're talking about here in terms of this oral loss of tolerance is what is fundamentally happening from the start that leads to this, what I'll call a skewing towards more IgE versus regulation that's healthy and accepting of these food products.
Yeah, I mean, there's still so little that we know about the immune system in the first few months of life. We know that there are some factors, some exposure factors that occur in utero. So there may be some increasing susceptibility already before birth. But then there appears to be a very important window after birth when the type of exposures matter a lot. And so just to get into an example, with peanut allergy, we know that, if you wait too late, then before you introduce it into the diet, you can develop peanut allergy. We think that exposure through the skin is a very important route where that exposure is driving the development of allergy. So this comes from a few, lines of evidence. Having eczema or allergic inflammation of the skin is one of the biggest risk factors for the development of food allergy, including peanut allergy. And we know that there is measurable amounts of food allergens actually present in household dust. So if you take dust samples from not just the kitchen, but a baby's crib, from a child's bedroom, you can actually measure detectable amounts of peanut protein in that household dust. So when that exposure combines with inflamed skin, that immune system that's present in the skin is going to react to all sort of foreign antigens as dangerous because it's in the context of inflammation. That immune system is already revved up and ready to react to exposures.
And hence the common link between skin and these GI issues, or in the case of food, specifically in food allergy, because those are both epithelial barriers. So people hear the word epithelial thrown around. Both of those are epithelial barriers and have different but similar mechanisms in play in terms of barrier loss, barrier protection, and also immune mechanisms for different purposes, of course.
Yeah, they both have this shared quality of having to decide what to be tolerant to and still being able to protect us against all sorts of foreign antigens. And what the immune system has to do is distinguish between foreign antigens that are dangerous and foreign antigens that are innocuous. And normally our immune system does a really good job of distinguishing those.
Mm-hmm. Mm-hmm. Okay, so going back to this whole notion that and really what we're talking about, I mean, when people have asked this question in the past, like what causes food allergy, there's always this common response from the scientific community. There's part genetic, part environmental. We're not really talking about the genetic part right now. And I don't want to get into epigenetics and the complexity there,
right.
But this is more the environmental part of it. What's changed in our world that may have contributed to more of these disease, just incidences in terms of the population, above and beyond kind of baseline genetics, if you will. Is that a okay way to describe that?
Yes, yeah, and in a way there are so many different things, and everyone has their pet theory about which one is most important. So, and I think all of them have valid scientific evidence underneath. So things like chemical exposures may play an important role. There has been description of things like detergents, laundry detergents, and dishwasher detergents affecting the barrier of the skin and our gut. So that's one theory. The other is that the environmental microbial exposures have changed just due to urbanization and the way that we live. And that leads to a change in the composition of our microbiome over time. There are changes in the way that we prepare our food, very sort of subtle changes in food processing that may influence our exposures. And so, there are many exposures. There are things like vitamin D, for example, is another one which has been hypothesized to play an important role. And how much vitamin D we're getting and through what sources may play an important role. And so,
stop, you just made it too complex.
Yeah.
Well, I thought we were just going to put people on farms and we'd fix the whole problem.
Well, I think the sort of good news is that there's not one thing that is completely driving food allergy. And the bad thing is that we don't have an easy fix, that we haven't yet found one environmental factor that could, if we withdrew it, wipe away food allergy.
Right. There's something funny about that comment you made about the dishwashing detergents or liquid, because we both know which researchers you're referring to. Yes. And it for the public's consumption, that's a couple in terms of their, it's a scientific research husband and wife powerhouse couple in this field of immunology. And they take that theory very seriously, because I know that they don't use soaps anymore and they don't run their dishes in the dishwasher anymore. So they hand wash everything with hot water and very light, diluted soap. So I think that's interesting that their own research is, they practice what they preach, I guess is what you say.
Yes. Well, I do think about that every time I unload my dishwasher.
So do I. And the film that they're talking about on your cups, I'm like, oh, ruining my barrier function.
Yeah.
Okay, so you actually mentioned two things it's a good transition. So you alluded to early introduction of food and kind of this notion that not presenting these antigens early in life was not the right kind of guidance that was given a long time ago. And that was more just, best theory at the time. It wasn't deliberate in terms of trying to obviously, delay, this presentation. I think this understanding of how this exposure occurs has been exposed over, the years of scientific research. Now that we know, and we have things like the LEAP study and ongoing studies that are going to push the early introduction of food. And if we don't make the obvious link, food is also currently the treatment for food allergies. So OIT therapy is technically food product, right? Given as a therapy. So in one case, you can prevent potentially. In another case, you could treat. And sometimes you can see some folks on OIT that get this outsized effect and get almost curative-like responses, or what I think we call sustained unresponsiveness.
Mm-hmm.
What's going on there? So how come in one case you can prevent, and in another case you get this outside response? In another cases, it's very temporary unless you continue maintenance therapy. So what do we know about that?
Well, that's a major goal of the research that's going on in my lab, as well as many other labs. One of the things that we're trying to do is study, who does well compared to who does not benefit so much from immunotherapy, to try and understand the underlying immune basis. Now, we think that age and maturity of the immune system plays a major role, because we do know that younger children seem to do better with both oral immunotherapy and epicutaneous immunotherapy. There's a lot of interest right now in trying to understand how young do we need to go and why. So is there a biomarker of a kind of hardened immune system that could tell us, okay, that may not do so well with allergen immunotherapy alone? Maybe we would need to add another kind of immunomodulator in order to get an effect. So that's, it's still something that is very much being studied, but I think likely has to do with this maturation of the immune system that occurs with age.
Yep, this whole notion of early life immune development, or the term plasticity, is thrown around quite a bit these days.
Yes.
And I think what's interesting about food allergy is, we've always talked about food allergy being so behind other areas of scientific research. Well, this scientific discovery or breakthrough that you can intervene early with just food and change the immune response, I think is just underestimated how powerful that is around what that conveys, because that really almost proves what you just said. Intervening early makes a difference.
Yeah. And I do think that it's, we've known about this concept of oral tolerance for a long time. We didn't really understand if it was the basis of health. And through studies which actually compared children who received peanut early in life or who didn't, that basically proved that the concept of oral tolerance really worked. And I think, for any disease, prevention is so much easier than treatment. And I think that we have, if we can do things that are not very invasive, not a burden, inexpensive, in order to prevent food allergy, that's really, I think, very exciting.
Absolutely. I can't wait to get there.
Yeah.
Next year, right?
Soon.
Soon. Okay. So we could just talk about this all day. I have so many other questions, but let's come back to one thing we talked about earlier. So IBD.
Yeah.
IBD is an autoimmune disease. And autoimmune diseases, the notion is that your body is creating self-inflicted harm on itself, right? You are presenting an antigen and your body is now reacting to that antigen as if it's foreign and causing that inflammatory response. I mean, very simplistically,
right.
It's kind of what's going on in these autoimmune conditions. Now, what happens in each one is obviously very specific and different, but generally there is, again, a loss of a tolerance there to something that is being presented. Is that the right way to describe that in autoimmunity and things like IBD?
Yeah, and I think what's interesting about inflammatory bowel disease is that um, the um, the loss of tolerance is also to something that um, is technically foreign and generally innocuous, and that is to our intestinal microbiota. And so there is this lack of tolerance. The mechanisms how we tolerate food versus how we tolerate our microbiota is a little bit different. They most of our exposure to foods comes higher up in the gastrointestinal tract than our exposure to bacteria. So there are some differences between those, but fundamentally it is a lack of normal, healthy immune tolerance and these regulatory pathways that are so important. And I think what we can learn from inflammatory bowel disease is that they've done much more with immunomodulatory treatments, things like biologics. They've been using anti-TNF and so cytokines, targeting cytokines and targeting different trafficking molecules more than has been done in food allergy. I think one of the things that the food allergy field can teach inflammatory bowel disease research is, we have sort of benefited from the fact that we know what's triggering these inappropriate immune reactions. We know how to be able to identify a particular cell that is, reacting to peanut, that is causing the generation of IgE antibodies. So we have this specificity, and now in inflammatory bowel disease, they're starting to, identify what are some of the microbial triggers, and we can now help that field try and understand, how to study those specific cells that are triggering disease.
You said it. You took the words out of my mouth, because that is I think the dream, that we get to a place where we do leapfrog enough, where we've advanced the science and we could not just learn from other areas, but we can help lead in some areas. And I really think that this early life loss of oral tolerance and understanding is such a powerful one, that food allergy can be, a scientific field of research that really unpacks a lot of what's going on there, because it absolutely is clear and compelling that, this is the way that our field of research will continue to progress.
Yeah, I think, one of the areas where, we have this real challenge is in this context of specificity. Someone may be allergic to peanut, but they're tolerating egg and wheat and milk. So all of those regulatory systems are in place for the other foods. It's very specifically about peanut. And that gives us tools to be able to really investigate, okay, in this one individual, what's a healthy response and what is an allergic response? I think it really helps us scientifically to have that specificity. And I think with specificity on the treatment side, I think there are 2 points. One, there's a need for understanding the specificity in order to treat, but there's also, many individuals are multi-allergic. And if we can come up with approaches that can target multiple food allergies, I think that would be, very powerful.
You need specificity, and then you need to find the common mechanism for broad spectrum therapy.
Yes. Yeah. So I think that the specificity will help us learn.
Yeah.
And then the application has to be broader.
But you're absolutely right, because I am familiar with the evolution of the immunomodulatory therapies in Crohn's disease and IBD. And that field, as it evolves, the next generation of class of therapies that are also coming in that space are antigen-based. So antigen-based therapies in combination with either antibodies or microbiome products or other mechanisms like you talked about earlier. Maybe you're going to need a couple of different things to get that full response.
We don't really quite know. And again, it's most likely or almost certainly heterogeneous in terms of the nature of that. Let's talk about treatment for a second. You've talked about immunomodulatory therapies. There's obviously OIT. What's in that landscape? And, throw in prevention as well, because obviously there's a lot of activity in prevention. What kind of interventions do you think are, this is such a generic question. Exciting is not the right word, but either from two perspectives, either are the next most critical class of therapies that will unlock the next, opportunity for therapy and intervention, or represent kind of very next generation approaches at treating. Take either flavor, but I think both would be interesting to hear about.
So I think the closest to actual clinical use is omalizumab, or targeting IgE antibodies. So this has been around for several years now. It hasn't really been applied to food allergy. There have been clinical trials that test it together with oral immunotherapy. But one of the big trials that's being done within this consortium for food allergy research right now is testing omalizumab both as a monotherapy and together with multi-food immunotherapy. So, and this trial will actually go a step further, and in the sort of third stage of this trial, there will be dietary introductions of foods. I think that this has the potential. So if omalizumab could be given, it blocks IgE, or it binds to the IgE antibodies, reduces them. It makes an individual much less reactive to those foods. If you could use that as a treatment, and under sort of the protection of that, introduce things into the diet, and then wean off omalizumab, it may be a very short-term use of omalizumab that then allows for dietary introduction and protection against foods. We don't know if it's going to work. We don't know if withdrawal of omalizumab will then lead to starting to have reactions again. So that's why it's a clinical trial. But I do think for multi-food sensitized individuals, it may be the next thing up. We know that IgE antibodies they are made in response to cells called T cells that are making cytokines. And the two cytokines that are really critical for making IgE antibodies are called IL-4 and IL-13. So there is a biologic, it's an antibody that binds to the receptor that's shared between IL-4 and IL-13. It's called Depilomab or Dupixent. And this is in clinical trials right now. It's being tested both as a monotherapy and together with oral immunotherapy. And the idea is, if you're giving allergen along with blocking the main driver of IgE production, you're going to be much more effective at reversing the IgE sensitization. Now, we know from other allergic diseases like eczema that if you stop giving this anti-IL-4 and IL-13, the immune response just bounces back. There's a question of whether that will also happen with food allergy. So if you give this blocker together with immunotherapy, would you now really modify disease to the extent that you don't need to keep on giving Depilomab? So again, that is being tested. There are a whole bunch of other biologics targeting different parts of that so-called Th2 pathway. And we'll see how that works. In mouse models, it works pretty well. So there's a question, there's the hope that will also be effective. And it's, this is a drug that has really, I think, revolutionized eczema treatment. So I think that there is a good chance that it will have a significant impact in food allergy. The third thing I want to talk about comes under the category of microbial therapeutics. And this, I think, is also very exciting. So there are many different companies that are in this, this area of trying to develop, I sometimes refer to it as a next generation probiotic. It's really, the approach has been quite different, that it's more evidence-based. So by studying individuals who are tolerant versus allergic, that particular microbes have been identified that are regulatory, or metabolites that, such as butyrate, have been identified that are regulatory. So there are clinical trials ongoing with microbial therapeutics plus oral immunotherapy to see whether or not, the idea is that if you give the right microbes back, it's going to change the immune tone of the gastrointestinal tract. That now when you start to get that oral immunotherapy, you're seeing that antigen in a much more regulatory environment. You're going to have a better outcome. So that's the idea. And there's also a lot of interest in using that as a preventive. So giving this early in life, can you give an appropriate microbiota and prevent food allergy? On a sort of related note, one of the studies that we're doing here at Mount Sinai, I mentioned that C-section birth is associated with a slight increase in the prevalence of food allergy. So on an individual basis, it's not like having a C-section will definitely make your child's food allergic. It's a very small risk, but on a population-wide approach, that has a big impact. One of the things that we're studying is whether or not exposing infants right after birth to the vaginal microbiota, basically by swabbing their face and their body with the vaginal microbiota that they're not being exposed to because they're getting a C-section birth, whether or not that will reduce the risk of food allergy. So, it's both, microbial therapeutics that have been developed, similar to a pharmaceutical therapeutic, but also, there are approaches, their management. So for example, should we be swabbing babies that are born by C-section? If that becomes a standard of care, can we reduce a certain fraction of food allergy that happens? Similar to, if we introduce foods into the diet at 4 to 6 months of age, if we have clear feeding guidelines for what foods when, can we knock down a big chunk of the food allergy that we're dealing with? And also, hopefully we can study those and figure out, okay, for those who can't develop tolerance, who haven't developed tolerance and who already established kind of a hardened allergy, what can we do for those individuals to change their immune system back to tolerance?
In all seriousness, you and I should probably talk offline at some point, given what you're doing. And it's been actually top of mind that I wanted to reach out to you at some point to follow up on these areas of scientific research, especially what you mentioned with the C-section study. And things like that are very interesting in terms of just, what is going on with that early life window of plasticity. And how do you think about low cost interventions that could really have outsized impact? That's just a tremendous opportunity scientifically. Thank you for that. I think that was a wonderful overview of the stages of different products in development. And I'd like to remind the audience that if you rewind the tape 5 years ago, and we're in 2021, if you go back to 2016, I believe at that point, Xolair had not even announced breakthrough status for food allergy yet. So we had only the immunotherapy products in clinical development and almost a very, very thin to barren landscape of other products. And if you look at the development pipeline now, fast forwarded 5 years from then, it's a different pipeline. And it's only going to continue to grow, I think, based on what you just alluded to around these overlapping opportunities and mechanisms in terms of drug therapies.
Yeah, I think that there's a much more going on now also with academia and industry collaboration, which is really going to help accelerate the pace of research.
That's what we talked about with Cynthia from JDRF, because obviously that is, what FAIR would love to accomplish, and is still the mission for everything that's been developed and ongoing. So again, kudos to that team and to keep pushing on this scientific discovery, because it does take time, but the amount of progress that could be made in the next few years is really exciting to think about. Okay, we are out of time, unfortunately. So I'm going to open it up for some questions from the audience, to the extent that any of our limited life, audience that has been listening in has any questions. We did get one, and I'll start with that one. So we talked about transient disease and this window of immune plasticity earlier. The question came here, just in terms of outgrowing an allergy. So this person's son has nut allergy, now in their mid-20s, was diagnosed at 4, but now can eat almonds and pine nuts, but is still allergic to all the other nuts. So what's happening there in terms of maybe an older population still having some ability to adapt or evolve, and what really happens, or what do we know happens?
Right. Well, this is a very interesting question. And, when you think about different foods, the classic example is milk and egg, which are commonly outgrown in the first few years of life. The allergies to milk and egg are commonly outgrown, whereas allergies to nuts are not commonly outgrown, but they are outgrown in some individuals. It's a minority of the group, but we know that it does happen. So with milk and egg, it's a little better understood. We know that the type of cooking, so for example, most kids who are milk allergic can tolerate cooked forms, extensively heated forms of milk. So we know that the actual binding of the IgE antibodies on the milk differs between those who can tolerate or not extensively baked forms of milk, and that the ones who can tolerate the baked milk are more likely to outgrow it. So we think that it's basically the sort of maturation of the digestive process and the handling of those antigens that allows the IgE that are recognizing only sort of what we call conformational epitopes. It's the structure of the protein before it's been denatured, that appears to be more transient. We don't yet understand. So for example, if you take a cohort of peanut allergic kids at age of 4, somewhere between 10 and 20% may outgrow their peanut allergy. So one of the things that we're trying to understand right now is how does that immune system response differ? Is it because they're recognizing different epitopes on the peanut protein, and their digestive processes mature with age, and that they change the way that they handle those antigens in the gut, and that leads to the development of tolerance? Or are there other factors? I think one of the most sort of exciting areas of research going on right now in food allergy is trying to understand what's the basis of IgE memory. So once you've generated an IgE response, how can you lose it? So why, if one child has an IgE response and they lose it, whereas another child has an IgE response and they keep it, and they're both very carefully avoiding peanut, they're not getting this repeated exposure, what's the immune basis of that? If we can figure out why one child loses their IgE memory and can then target that in everybody else, I think that that would be fantastic. So that's our goal, and there's a lot of people who are actively working on that right now.
I love that you raised memory. I wasn't going to mention it, but I've listened to Jerry Niepom from the Immune Tolerance Network talk about these frameworks for immune tolerance quite, I've seen him present this framework a few times. And, oftentimes, and these are findings from type 1 and other areas, that if you don't solve the memory problem, you're going to maybe have a durability problem with your therapeutic response. So maybe we can get these mechanisms all to work. And in some percent of the population, they may have this tolerant effect, and others maybe not. And to what degree do these memory cells have a role in kind of, your inability to lose your immune response, good or bad? I think it's a fascinating area of research, because the
thing about those cells is they're just hard to find and they're not abundant. And
Yeah.
understanding what they're doing and how they work, I think, is a really interesting area of research. That's
Right.
I mean, and people may be wondering right now, what are these people talking about? Your immune system has a memory? More or less, yes, because you have very specific cells that have these functions to bring back these responses, which is why in even viral diseases as well, it's
that's
hard
why
to
vaccines work, you know, and some
right,
vaccines
right.
last for life and others you have to keep boosting them. And it's all related to this concept of memory.
Perfect. Exactly.
Yeah.
Right. What you've communicated, at least in terms of that future landscape, is thrilling because you've talked about the nature of this heterogeneity, the sub segments of this population that we can learn from across populations of disease, across within the same patient type. And also this transient nature clearly seems to play some role in this evolution of our immune system. And so everything that's early life versus later life, and looking at all these comparisons of what is going on to basically unpack, you know, for more or less the secret sauce of immune regulation and immune tolerance is, again, it's an explosion that I think that will be fruitful in terms of intervention. And it feels like we're well on our way and, you know, starting to develop the right frameworks to think about how to unpack all of that.
Yeah, I do think it's really exciting. And you know, you just have to look at what's happened in the cancer field with their immunomodulation stories and checkpoint inhibitors. And, you know, that's really shown us the amazing capacity of what modifying the immune response can do. I think one of the things that it's also taught us is that there is, you know, the immune system is a very complicated beast, and that, you know, there is also some caution that needs to be taken with immunomodulation, that you need to really understand what is the downstream implications of this on the whole immune system. So I think as researchers, we have to, you know, strike that balance between being bold in what we're trying to do and being, you know, cautious. And that's a hard, it's a hard line.
Well, if people think of the immune system as a, you know, a self-regulating piece of machinery, then it has a constant, you know, it's in perpetual motion, right, over our lifetime, because it's always trying to regulate itself. And so just doing one thing, you can have other consequences, and you can't just overdo one thing. And that's why you get this skewing potentially of immune response. I mean, in the case of antigen presentation, you have this outside skewing towards this innocuous antigen of food. I feel like there's a lot of, you know, yes, medicine is incredibly powerful and often very targeted around one pathway, but also we have to understand that this is a whole system that works together. And, you know, thinking about the complexities and understanding the, you know, the ups and downs of that system and how it regulates itself is complex. And then how it differs is complex. So, you know, that's where the appreciation of, like, just, there's not a simple answer in these immune conditions, but there are clearly ways that you can peel back that onion and start to figure out, you know, the different, you know, approaches that you could take, you know, in different individuals and subtypes of populations.
Yeah. Yeah. And I really think that with everything that we're going to be trying, we need to be carefully studying those who do well versus those who don't benefit, and really figure out what it is specifically about those who do well that you know, is responsible for that benefit.
All right. Well, this has been a fun conversation, at least for me.
And for me.
I hope it was, you know, educational and interesting for the people listening in and that will be watching this in the future. Before we drop, Cecilia, anything that you'd like to just share with the fair audience as we say goodbye?
I'd just like to express appreciation for the whole community for participating in the research, either through participating as a study participant or through support. You know, we could not do this without you. So it's really critical that we have we have support from the whole community.
Well, that's well said. And I echo that sentiment on behalf of food allergy families and individuals, being a father of a food allergy son. And I'd like to, turn it around and say thank you to you. You're obviously, people know Mount Sinai and the Jaffe Institute, and what kind of leadership you've provided historically as an institution, but also as an individual. And you absolutely are, helping to lead this field of research. So we turn it around and say thank you to you as well for the contributions. So keep it up. Looking forward to it, and really appreciate everything that you've been doing, and including your time today. Really, thank you for your time.
It's been a pleasure. Thank you.
All right, thank you, Cecilia. Take care. To the FAIR audience, thank you everyone for listening. We have a lot of exciting conversations coming up. We'll be talking to experts in the anti-IgE field, as well as other areas related to prevention and treatment of food allergy. So until next time, thank you.